Hello everyone, my name is Sanket Desai. I'm from Dr. Amit Dutt Laboratory, ACTREC, Tata Memorial Center. I'll be presenting a poster titled A Computational Approach to Study the Dynamics of Variability in Infectious Pathogens. At the outset, I would like to thank the organizers for providing this opportunity to present as well as for awarding fellowship. I'm very grateful to them for this. As we know, the pandemic has hit us hard and it has caused more than 10 million cases worldwide and also caused more than 0.5 million deaths worldwide. And these numbers are true till June 2nd. And no proven treatments means the only defense is screening or finding out novel approaches of detection and then contain the individuals. So in this particular area, our lab has contributed by developing a RT-PCR based assay and automated analysis pipeline for SARS-CoV-2. And also we have explored a Raman spectroscopy based detection of RNA viruses from human saliva. Both these have been accepted for publication currently. In addition to the conventional techniques used for screening, we know that NGS technology is also coming up as a useful tool for studying infectious. Now, adoption of NGS techniques is for pathogen analysis is not a new concept. Since 2014, it has been implemented in the Advanced Molecular Detection Program, which was launched by CDC, and it forms the core of that program. The routine diagnostic techniques have been complemented with sequencing and informatics analysis, which aid in clinical decision making further. A typical a typical NGS analysis workflow starts with sequencing of DNA or RNA using the platform of our choice, and followed by the analysis using various tools. Now the main bottleneck is that the analysis platforms, they depend on the kind of data set generated by the sequencing platforms. So the heterogeneity is the first step of bottleneck. Our lab has been working in the pathogen detection area with respect to tumor biology, in which we've developed HPV detector for detecting HPV from cancer samples. The same methodology was further developed to identify non-typhoidal salmonella, which is a bacteria from gallbladder cancers. Building on this concept of computational subtraction, we go further to develop infectious pathogen detector, which is extended for the SARS-CoV-2 datasets. So, in short, it is an integrated platform. IPD that we call is an integrated platform for quantification and variant analysis. It is a tool based to handle heterogeneous data, that is long and short read data both. It is developed mainly for the biologists with minimal computational know-how. So the NGS data is churned through the tools by using our custom databases at the backend, and it generates a user-friendly report for the biologists who will be working on the NGS data. So the database mainly consists of the infectious pathogens from FDA Argos as well as literature. And the database is divided into primary and secondary database that I'll be showing in the coming slides. The primary database is consist of both viruses and bacteria, which is composed of 1060 in total distinct species. So as I said, IPD is built for heterogeneous data. So the short read analysis it starts with filtration, followed by alignment to both human and pathogen genomes. Similarly, the long read data, which is mainly focused on nanopore right now, has two components, which is filtration and alignment, both giving us the pathogen reads are further quantified using our quantification pipeline, which also involves ambiguous read filtration, which is an important step, and the count normalization, which is based on the per million reads. The variant analysis pipeline starts with pre-processing and further consensus variant call are generated, which are annotated using the annotation tools. The long read analysis also follows similar pipeline in which we use additional long read specific variant caller. So benchmarking our tool for quantification, we see a correlation with GATK PathSeek and Kraken with 0.96 and 0.94 R value respectively. And this have been performed on the in-house oral transcriptome samples. We validated our tool, the quantification by our tool for Fusobacterium nucleatum, which is a commensal inflammatory bacterium using qPCR. And we find a lot of concordance between qPCR and our tool, as well as quantitatively it shows distinct pattern with respect to. Hence, we further go ahead with analysis of more than 6,000 samples, long and short read samples. Here I'm showing a snapshot or a shortened version of the analysis of transcriptome, in which we show specificity towards the SARS-CoV-2 and MERS separately, and the negative samples being in-house oral transcriptome samples. The IPD is also developed for a variant analysis, in which at the back end we have generated a variant database based phylogenetic assignment algorithm. Mainly works on the VCFs which are generated by IPD, and they find the closest relative to the isolates which are available in the database, and then the node assignment happens, which can be distinct A, B, or C node. Depending on whatever output comes, we present it in a form of a report, in which basic sequencing statistics is provided, followed by quantification and coverage of the pathogen, and the novel and the known variants are also reported. So this becomes a very important tool in the current pandemic, and it gives an overall picture of the sequencing that has gone into the pathogen sequencing that has gone into, as well as the evolution in a form of phylogenetic analysis of the variants. With this, I finish with the acknowledgements to the funding, that is Tata Memorial Center, and my guide, Dr. Amit Dutt, the IPD team, which is Sonal, Aishwarya, and Bhaskar, and my lab members for all their support.